For many years, it was widely believed that only proteins could be enzymes. An enzyme must be able to bind a substrate, carry out a chemical reaction, release the product, and repeat this sequence of events many times. We now know that RNAs can also act as biological catalysts. RNA molecules that act as enzymes are called ribozymes. When you have completed this exercise, you should understand the structural differences between DNA and RNA, understand how these structural differences help RNAs act as catalysts, understand the catalytic activity of the Hammerhead ribozyme, and be aware of some of the other catalytic activities exhibited by ribozymes. While the structures of RNA and DNA are similar, RNA differs from DNA in three key respects. First, RNA is usually found as a single polynucleotide chain, rather than a double strand. This allows RNA to fold back on itself, forming complex tertiary structures. Second, RNA contains uracil in place of thymine. Uracil is identical to thymine, except that it lacks the 5' methyl group. Third, the backbone of RNA contains ribose rather than 2' deoxyribose. The substitution of a hydroxyl group for a hydrogen atom is important for the catalytic activity of many ribozymes. Under alkaline conditions, the 2' hydroxyl of the ribose in the RNA backbone can become deprotonated, resulting in a negatively charged oxyanion. This oxyanion can attack the phosphate group at the 3' position of the same ribose. This reaction breaks the RNA chain, producing a 2' 3' cyclic phosphate and a free 5' hydroxyl group. Each ribose in an RNA chain can undergo this reaction, completely cleaving the parent molecule into nucleotides. The Hammerhead ribozyme is a sequence-specific ribonuclease, whose biological activity uses a mechanism similar to alkaline hydrolysis. The Hammerhead ribozyme was originally discovered in certain infectious RNA agents of plants called viroids, and has since been discovered in the genomes of species ranging from newts to cave crickets. The Hammerhead ribozyme gets its name from the shape of its secondary structure, which consists of 3 base-paired stems surrounding a core of non-complementary nucleotides required for catalysis. The Hammerhead ribozyme cleaves RNA between stem 1 and stem 3 at cellular pH. This cleavage is accomplished using a magnesium ion found in the catalytic pocket. The magnesium ion coordinates a hydroxide ion in an optimal position to deprotonate the 2' hydroxyl of the ribose at the cleavage site. Following this deprotonation, the mechanism is identical to the mechanism of alkaline hydrolysis. In the tertiary structure of the Hammerhead ribozyme, the cleavage site is positioned within the catalytic pocket. This orientation is necessary so that the magnesium ion bound in the catalytic pocket can catalyze the cleavage reaction. Note, this animation is meant to aid you in understanding the tertiary structure of the Hammerhead ribozyme. It is not meant to represent the biological mechanism of ribozyme folding. Naturally occurring Hammerhead ribozymes are autocatalytic, and the self-cleavage reaction can only take place once. Therefore, naturally occurring Hammerhead ribozymes are not true catalysts. However, the Hammerhead ribozyme can be engineered to function as a true catalyst by dividing the molecule into 2 portions, separating the ribozyme from its substrate. Like the hammerhead, many of the known ribozymes are ribonucleases. However, ribozymes have been discovered with other activities. For example, the component of the ribosome that is responsible for the formation of the peptide bond, the peptidyl transferase, is a ribozyme. Researchers have used in vitro evolution to generate ribozymes that catalyze other reactions. In this exercise, you will explore some of the techniques used to evolve new ribozymes. The discovery of ribozymes led to the RNA world hypothesis. According to this hypothesis, there was once a form of life based entirely on RNA, without proteins or DNA. For this to be true, the RNA would have to have a way to replicate itself. If such an RNA polymerase ribozyme could be created in a lab, this would lend support to the RNA world hypothesis. For this experiment, you start with the catalytic core of a previously discovered ribozyme, which is capable of ligating stretches of RNA, but not of copying RNA from a template. You add a random sequence segment to the 3' end of the starting ligase to allow for the evolution of a new catalytic domain. You must now create a pool of RNA sequences. Question 1. Of the techniques listed, which would make a good first step toward creating a pool of RNA sequences? Question 2. Of the techniques listed, which should you use to generate a large pool of different DNA sequences? Question 3. Of the techniques listed, which should you use before testing for ribozyme activity? Now that you have a pool of different RNA sequences, you must select for the desired activity. Since copying of nucleic acids often requires a primer sequence to jumpstart polymerization, you covalently attach a short primer to the 5' end of the ribozyme sequences. You then incubate the RNA sequences with a stretch of template RNA, as well as nucleoside triphosphates. You are hoping that some RNA molecules in your pool are capable of extending this primer. Ask yourself, how will the newly synthesized RNA be different from the template RNA? You can use gel electrophoresis to select for RNA polymerase activity. Question 4. Select the part of the gel that contains RNA sequences with RNA polymerase activity. Question 5. Place the experimental techniques in order on this diagram of the in vitro evolution cycle. You were able to pull a few RNA polymerase ribozymes out of your RNA pool, but their activity is very low after only one round of selection. To evolve ribozymes with greater activity, you must repeat the mutagenesis and selection steps several times. Question 6. You are trying to evolve an RNA polymerase that can replicate any RNA sequence. What could you do to make sure your polymerase does not only recognize one specific sequence? After 10 rounds of selection, you have an RNA capable of catalyzing the replication of an RNA template. However, this RNA is not technically a ribozyme, since it can only make one copy of the target RNA. Question 7. How can you turn your RNA into a true ribozyme? The structure of RNA differs from that of DNA in 3 key respects. RNA is usually single-stranded. RNA contains uracil instead of thymine, and RNA contains ribose instead of 2' deoxyribose. These differences help RNAs to act as catalysts. Under alkaline conditions, the 2' hydroxyl of a ribose in the RNA backbone can become deprotonated, leading to the hydrolysis of the RNA chain. The Hammerhead Ribozyme catalyzes the hydrolysis of RNA in a mechanism similar to that of alkaline hydrolysis. Ribozymes have been discovered with several activities. For example, the peptidyltransferase of the ribosome is a ribozyme. You have completed this exercise.